Hey, in the last video, we started to look at nucleic acids by discussing some aspects of nucleotides as the building blocks of nucleic acids such as DNA and RNA. In this unit, what we are going to focus on is how those individual nucleotides link together by covalent bonds in order to yield a strand of DNA or a strand of RNA. So let's take a look at that chemical process here as we continue our journey of building the foundation for chemical biology. We'll go ahead and get the projector going here to screen share. And the handout that you will see on the screen here is the same one that I used in the last video. You can access that under module number 1 to follow along with this so that you don't have to redraw quite everything. I have also gone ahead here on the screen and drawn in the chemical structures that we're going to be working with as we try to answer this central question of how do nucleotides link together to yield DNA or RNA as a strand. So we need to be able to absolutely do this in order to generate DNA and RNA. So we have our building blocks here. We've drawn a couple of nucleotides here and here. You'll notice that one of these has a triphosphate group on it, which is a bit distinct from the monophosphate group that we originally saw in the lower right-hand corner when we were talking about nucleotides. This modification is going to facilitate the formation of the linkage between the two nucleotides, the nucleotide in pink and the nucleotide in green. The reason is that this triphosphate group stores a lot of energy. And when we start breaking these phosphate oxygen bonds, that is going to release energy that will drive this chemical reaction. This particular chemical reaction of linking nucleotides together within a cell is catalyzed by an enzyme known as ligase. So ligase is what is going to catalyze the stitching together of the pink nucleotide and the green nucleotide. Specifically, the type of bond that that enzyme, ligase, is going to catalyze formation of is referred to as a 3,5 phosphodiester bond. The reason why we refer to it as a 3,5 phosphodiester bond is because we are taking carbon number 3's hydroxy group and linking it to the phosphorus atom that is bonded at position 5, carbon number 5. So hence a 3,5 phosphodiester bond. We refer to it as a phosphodiester bond because what we are going to see after this process takes place is that we are going to have the phosphorus group here, still bonded to a carbonyl and bonded to two oxygens that then have carbons flanking on both sides. So keeping in mind that an ester functional group has a carbonyl group directly bonded to an oxygen, directly bonded to another oxygen, and a diester would have a second oxygen there and second carbon. We could think of this phosphodiester as being analogous, except that we have a phosphorus group, phosphorus atom rather, in place of the carbon atom. So in the case of a diester, we have a carbon atom here as part of a carbonyl. Here we have a phosphorus, hence phosphodiester. So that is the type of linkage that we're going to be creating in our final product here as we draw out this molecule and think about the mechanism to answer that question of how these two molecules are linking together via covalent bond and ultimately linking to others so that we could form a strand of thousands or millions of different nucleotides linked together to form a DNA strand or to form an RNA strand. So the reaction that is going to take place here is a situation of nucleophile and electrophile chemistry. Now, the phosphorus atoms that are in our structure here at position 5, this phosphorus atom, for example, has a very strong positive polarization that we see right here. It's positively polarized because it's bonded directly to four free electronegative oxygen atoms that are going to be strong electron withdrawing groups. So that makes the phosphorus atom very strongly positively polarized, meaning that is going to make that phosphorus atom electrophilic. On the other hand, in this enzyme catalyzed reaction, we have an oxygen atom here at position 3 of the nucleotide that I've highlighted in pink. And that oxygen atom is really well set to act as a nucleophile. And so with the ligase bringing together these two nucleotides and positioning and poising them for reaction, what is going to happen is that the nucleophilic oxygen atom attacks the electrophilic phosphorus, and one of these oxygen atoms has to break away. The one that's going to be most prone to break away to yield a stable product is going to be breaking those additional two phosphate groups away. So rather than this being a triphosphate group, boom, once we break that away, we're going to be left with one phosphorus atom in our organic product of this reaction. So let's go ahead and draw out that organic product. And I'm abbreviating in all these structures in base, referring to the different nitrogenous bases that can appear in our different nucleotides. So that would be any of these possibilities in the upper right-hand corner is what could be filled in as the N base there. I'm just trying to simplify things in our structure a little bit so that it doesn't take quite all day to draw it out. We'll leave position 3 alone in this structure, that following the green structure there. Coming up to position 5, we have our methylene group CH2, oxygen, phosphorus atom with the double bond to oxygen. Then we still have that oxygen anion down below. We come over, and at that point, we're linked to the oxygen atom. And so I'm going to highlight all of these atoms in green are what originated from the nucleotide in green up top here. And now we can do some pink highlighting here, because this oxygen atom that I've highlighted in pink down in the lower part of the screen is the oxygen atom that acted as the nucleophile here, using that electron pushing arrow to show the formation of a covalent bond to the phosphorus atom. So now we're into the oxygen, and that connects to the rest of this molecule that we showed in pink in the reactant. So I'm going to go ahead and draw that out here. And of course, as usual, I'm running woefully low on room here. So I'm going to just erase a little bit of information there to squeeze in our reaction product. So our reaction product now is going to have our 5-membered ring, our nitrogenous base still there, and our CH2 bonded to our phosphorus, which again, running woefully low on room is kind of my jam. So we'll go ahead and plug in that phosphate group PO3 with a -2 formal charge there. And to finish this off, I'm just going to highlight in pink the rest of the parts that came from that nucleotide up top that's highlighted in pink as well. So our final product is going to be this structure here, which I'm highlighting with my laser pointer right now. And the key thing is that we've linked the oxygen here that originated from our sugar molecule up top at the 3 position to the phosphorus atom of the second nucleotide to form this so-called 3,5 phosphodiester bond. And it's a 3,5 bond because it originated from position 3 of one of the sugars, and it links to the phosphorus atom that was at carbon number 5 of the other sugar molecule to make this phosphodiester bond. And then what would happen in continuing this process of constructing a DNA or RNA molecule is that we would continue down the chain here, linking together time after time after time to create a strand of DNA or a strand of RNA that was comprised of a very specific series of bases comprised of the adenine, guanine, cytosine, and thymine if we're talking about DNA. This strand can be up to millions of nucleotides inside. So this reaction can occur literally millions of times within a single cell, which is pretty wild and amazing if you think about it. So what we will get into in the next video segment is we're going to talk about, once we've assembled a strand of nucleic acid, how is that strand of nucleic acid going to interact with another strand of nucleic acid, specifically by hydrogen bonding. So stay tuned for that.